continuation class of AIDS to Health. Today we will learn about toxins, antitoxins, antibiotics and antiseptics. Okay, so what are toxins and antitoxins? So toxins means poisons, right? So what are toxins? Toxins are general term used for any poisonous substances produced by microorganisms, by bacteria or by an animal or a plant. Yeah, so toxins generally, in general term, we should call it poison, which is very harmful chemical substances. Yeah, so whenever the microorganisms enters our body, that microorganisms will produce the toxic chemicals, or we can say toxins. Alright? Now, a few general toxins, you can look over here. The venom, the poison, the snake, the sting poisons of scorpions or the insects. These are all the toxins, which are very, very harmful chemical substances. Alright? Now, toxins are also released by the pathogens, the microorganisms growing inside the body. So when the bacteria, when the viruses, they enter our body, then they will also produce the toxins inside our body. And in that case, our body will produce the antitoxins. So what are antitoxins? Anti means against. Okay, the word anti, A-N-T-I, anti means against. Toxin means those harmful chemical substances. So, in respect to toxins, our body will produce the antitoxins. We have our immune systems. Clear? So, what are antitoxins? Let's go here. Antitoxin was the name given to any chemical substance produced inside the body in response to the entry of foreign poisonous substances. For example, let's say whenever the bacteria or viruses, when they enter our body, they start producing the toxins, the chemical substances which are harmful for us. Now our body immune system will come into action, so it will produce the antitoxins. So these antitoxins will work against the toxins. Clear? So antitoxins are those chemical substances which are produced inside our body in response to the toxins. Alright, which are released in our body by the foreign microorganisms. Now, antivenoms or antivenom. See carefully, antivenoms or antivenoms are usually given for snake bite, which are produced inside animals like horses. That means in this case, we are using the ready-made antibody which are produced in other animals. And if you remember, try to recall that particular conditions in which we will take the ready-made antibody. Remember the passive immunization or passive immunity. In passive immunity, we used to take or administer the ready-made antibody which are produced in other animals. For example, in case of snake bite, we used to take antivenom, right, or antivenin. Those antivenom or antivenin are produced in case of horses, and we used to take those antivenom. So an example of passive immunity. Yeah. So antitoxins are mainly produced in our body in response to the toxins. Alright. Now next one. What is antiseptic and disinfectant? So antiseptic and disinfectants are mainly used to prevent the diseases. So what are antiseptics? They are the mild chemical substances which when applied on the body kill the germs. Alright, we used to apply the antiseptic on the skin. So whenever we used to apply the antiseptic in our body, they generally kill the germs. But in your examinations, we don't write down examples of the antiseptic like Dettol, Savlon, alright? But they contain the active ingredients. The presence of active ingredients make them to function as antiseptics. So kindly don't write down Dettol, Savlon like example of antiseptics. Clear? So what are examples of antiseptics? We can see over here. Iodine. You can see over here. Betadine. That is iodine. Okay, benzoic acids, boric acids, boricate. Antiseptic. That contains boric acid. Mercurochrome. You can see over here. Okay, carbonic acids. These are the examples of antiseptic, which are the mild chemical substances which, when applied on the body, kill the germs. But there's a precaution that you have to take while using the antiseptics. Clear? Now look over here. Antiseptics and disinfectants are the different terms. Alright, they have different functions. So what are disinfectants? Look over here. They are the strong chemical substances that are applied on the spots and places where germs thrive and multiply. Antiseptics were being used on the body, on the skin parts, to kill the germs. They are the mild chemical substances. But disinfectants are applied on those places, on those spots where germs can thrive, where germs are present and where they can easily multiply. We are not applying the disinfectant directly on the skin or the body. We have to prevent our skin from contamination from those disinfectants. They are very strong chemical substances. That's why look over here. All disinfectants are strong and should not come in contact with the human skin. So the precautions of using the disinfectants. Why? They are strong chemical substances that are applied on the spots and places where germs can thrive and multiply. So avoid the contact of our skin, or the body skin, with the disinfectant. They are strong chemical substances that are mainly applied or used on the spots or on the places where germs are present and where they can multiply. So what are the examples of disinfectant? You can see over here cresol, okay, phenol, then you can say Lysol, okay, then you can say 40% formalin, and DDT means dichlorothiophenol trichloroethane. Alright, don't remember the abbreviation, just remember DDT. DDT is a disinfectant. Okay, DDT is a disinfectant. And remember, these are examples of disinfectant. So now we will discuss the difference between disinfectant and antiseptic. Before that, let's look at the other examples of disinfectant. Look over here. A strong heat and boiling can also kill the germs. Alright, and that's what they are called. Physical disinfectant. So heating and boiling can kill the germs. Second, look over here. Deodorants, which we used to apply. It is neither an antiseptic nor a disinfectant. It's just simply used to mask the bad smell. So deodorants are neither the antiseptic or the disinfectant. So kindly note it down. Okay. So what are the differences between antiseptics and the disinfectants? Antiseptics are the mild germ-killing chemicals, whereas disinfectants are the strong germ-killing substances. Antiseptics causes no harm to the skin and the body because they are mild, whereas disinfectants, they make us harm to the skin and the body. So we have to avoid the contact with the human skin with the disinfectants. Third point, antiseptics applied on the body. Alright, what disinfectants are applied on the spots, on the places where the germs can thrive and where they can easily multiply. So example of antiseptics are iodine, they are going to go through you, and disinfectants are phenol, cresol, Lysol. Clear? Now, next slide. Antibiotics. What are antibiotics? Antibiotics are chemical substances produced by certain microorganisms. And remember, antibiotics are chemical substances, and these substances are produced by certain microorganisms like bacteria that can kill or inhibit the growth of other microorganisms. Listen carefully. So antibiotics are produced from certain microorganisms. It can be from fungus also, okay, or fungi, even from bacteria that will kill or that will inhibit the survival of other microorganisms. So we'll go in details. The first antibiotic, which is over here, was the penicillin. So you can see over here antibiotics here. So these are those chemical substances which are produced by certain microorganisms that can kill or inhibit the growth of other microorganisms. So the first antibiotic was penicillin. which was discovered by Alexander Fleming in the year 1929. Okay, so Sir Alexander Fleming discovered the first antibiotic in the year 1929. The first antibiotic was penicillin. The source was afterwards. But the use of this antibiotic came into existence nearly in 1940. So in 1940, it was used first for the treatment of human diseases, and therefore the term antibiotic was coined by Salman Waxman in 1942. So you can see the example here, penicillin, the first antibiotic. Now, discovery of penicillin. So who discovered penicillin? Alexander Fleming discovered the first antibiotic, penicillin. So now, listen very carefully. So how did Alexander Fleming discover penicillin? So Alexander Fleming was walking on his laboratory on a bacterium called Staphylococcus. Okay, so mainly in the laboratory, Alexander Fleming was walking on a bacterium Staphylococcus. Now listen very carefully. Yeah. So let's say you have to know the two culture plates. So this is the first culture plate and this is the second culture plate. So you can see over here. This is the culture plate. Okay. So you're working on the two culture plate. This is the Staphylococcus. Here. Now you observe that. So there are two culture plates. First culture plate, second culture plates. Both the culture plate contain the bacterium Staphylococcus. Here. Now Fleming observed that in one of the culture plate, the growth of Staphylococcus was inhibited, means stopped. Okay, so let's say out of the two culture plates, so let's say in this culture plate, the growth of the Staphylococcus was not there. Clear? Whereas in other culture plate, it was growing normally, but in the second culture plate, the growth was inhibited. It was not growing, not multiplying. So Fleming observed that culture plate very carefully, and he observed that a certain mold, M-O-U-L-D, mold was growing over there. Okay, and the secretions of that mold was inhibiting the growth of bacteria. Yeah, a mold was growing in that culture plate, and the secretions from that mold was inhibiting, was stopping the multiplication of Staphylococcus, of bacteria. Clear? After certain time, Fleming observed that the secretions that was inhibiting the growth of Staphylococcus in the culture plate was mainly the antibiotic, and he named that as penicillin. So therefore, penicillin, the first antibiotic, was obtained from the fungi called Penicillium notatum. So this is the fungus Penicillium notatum, and from this fungus, the first antibiotic penicillin was discovered. Okay, so look over here. So Alexander Fleming was growing laboratory cultures of a bacterium Staphylococcus, which are the round bacteria growing in clusters. In 1928, he found that a certain unwanted mold of penicillium had grown in one of the culture. That means in one of the culture plate where there was no growth of bacteria, the mold was growing. The mold of penicillium was growing. Alright. It inhibited the growth of bacteria by secreting certain substances. So that mold of penicillium was releasing certain chemicals, and that chemicals was inhibiting the growth of bacteria in the culture plate. So Fleming named this bacteria-killing substances as penicillin. So penicillin is the first antibiotic which was obtained from the fungus called Penicillium notatum, and it will interfere with the multiplications of the bacteria in colony. Clear? So finally, go through it. Okay, so this is the Staphylococcus, the culture plate on which Fleming was walking. Now in this culture plate, the growth of bacteria was not there, it was inhibited. Why it was inhibited? Because in this culture plate, a mold of penicillium was there, and those penicillium was secreting a chemical that was inhibiting the growth of Staphylococcus. So plain name that particular chemical obtained from penicillium as penicillin. So penicillin is the first antibiotic obtained from Penicillium notatum. Clear? Now, penicillin was found to be effective against several infections. And here there are very good results in treating gonorrhea. Gonorrhea is a sexually transmitted disease. Pentyl sulfa, a combination of penicillin and sulfa drops. Sulfa drops means those antibiotics which contain sulfur. Examples of other antibiotics are streptomycin. Penicillin was the first antibiotics, but the other examples of antibiotics are streptomycin, chloromycetin, azithromycin, and penicillin. So these are the examples of few antibiotics. Clear? Next one. Sources of antibiotics. So over here, remember the name from where the antibiotics are obtained. So penicillin is obtained from Penicillium notatum. Whenever you will write Penicillium notatum, you have to underline it. Clear? Because it is a scientific name. Alright? So penicillin is obtained from Penicillium notatum. Similarly, streptomycin antibiotic is obtained from the bacteria called Streptomyces. You have to underline the word Streptomyces in the scientific name. So from Streptomyces, the bacteria, which antibiotic will get? Streptomycin. Now antibiotics are also known as synthetically. That means it can be synthesized in laboratory. Now penicillin was obtained from Penicillium notatum. That means naturally from Penicillium notatum we are getting penicillin. In a natural way we are obtaining it from Penicillium notatum. But nowadays we can also synthesize antibiotic. And when we synthesize antibiotic, not from the natural sources, but when we used to synthesize it in laboratory, then we should call it synthetically. Okay, next one over here. Commercial production of penicillin has been largely done from a related species called Penicillium chrysogenum. Now commercially also we can obtain penicillin from Penicillium chrysogenum. Clear? But first it is obtained from Penicillium notatum. So remember the source of antibiotic. Penicillin was obtained from Penicillium notatum. Commercially we can also get it from Penicillium chrysogenum. Streptomycin antibiotic is obtained from the bacteria Streptomyces. Now next thing, antibiotics can also be synthesized in the laboratory, which is called synthetically. Clear? So these are the sources of antibiotics. The next one. Uses of antibiotics. They are used to fight infections. Number 2. Certain antibiotics are also used as food preservatives. They can be used for the preservation of foods, mainly the meat and the fish. Okay. They are also used in the treatment of animal feed to prevent internal infections, and they are also used to control the plant pathogens, which can cause different type of disease in case of plants. So these are the uses of antibiotics. Clear? So remember the name 4 uses. First use, second, third, and fourth. So kindly remember the 4 uses of antibiotics. Next one. How does the antibiotics will help in the prevention of growth of bacteria? Because we are saying that penicillin, the first antibiotic, will inhibit the growth of bacteria, Staphylococcus. But how does it will work? It will come into actions. So mainly, these antibiotics will interfere by inhibiting the formation of cell membrane in bacteria or by destroying the cell membrane in bacteria. So let's see it. So penicillin interferes with cell wall formation. So the penicillin, the antibiotic, it will interfere with the formation of cell membrane in case of bacteria, which are unable to grow and multiply. Number 2, streptomycin binds to the bacterial ribosome. So streptomycin, the second antibiotic, antibiotic, it will go and bind with the ribosomes, and remember, ribosomes will help in the formation of protein. It will help in protein synthesis. So streptomycin will go and bind with the bacterial ribosomes, which are then unable to synthesize proteins, and the bacteria are unable to grow and multiply. So penicillin will interfere with the cell wall formation or cell membrane formation in case of bacteria, and streptomycin will go and bind with the bacterial ribosomes that will interfere with the synthesis of protein. By this way, the antibiotics will interfere with the multiplication or the growth of bacteria. They will prevent the multiplication of bacteria. Okay, with the actions of antibiotics, how will they kill the germs? Or the chemicals that contain sulfur. They are called sulfonamides. The sulfur containing drugs or the chemicals. Okay. So first of all, mainly in 1910, a drug called salvarsan that contains mainly arsenic was used for the treatment of STD called syphilis and sleeping sickness. Here, so these two diseases was treated with the help of the drug called salvarsan that contains arsenic. Then in 1930, a second group of chemicals was discovered that contains the sulfur group known as sulfonamides. Clear? So sulfonamides means that contains the sulfur group. And what example of sulfonamides? Sulfadiazine and sulfanilamide. These are the two examples of sulfonamides. These are the drugs that contain the sulfur. They mainly interfere with the metabolism of bacteria and lead to kill the bacteria. Then, nowadays, sulfonamides are rarely used, and if they are used, they are mainly used in combination with the antibiotics. Clear? So today we have learned about toxins, antitoxins, then disinfectants, antiseptics, then antibiotics and sulfonamide groups of drugs. So in the next class, we'll begin with the new chapter. Thank you.